Saya harap tak adalah gangguan-gangguan bising-bising. Okay, sebab last week hari Khamis, Jumaat tu saya buat kelas dekat rumah. So macam bising lah. Kan bising, so sebab saya datanglah college. Alright, so I'm going to share my screen. Okay, today we're going to discuss chapter 3. Okay, chapter 3. As usual, as I said in my lecture, you already learned this. Okay, this is revision for you. Okay, this is just revision. But it's okay, we're going to discuss this like we never heard before. Okay, topic 3, cell division. Okay, topic 3, cell division. If you have your lecture note, okay, if you have lecture note, you can refer to your lecture note, okay, or you can refer to mine. Okay, so these are the subtopics. We have the cell cycles, mitosis and meiosis. If you refer to the planner for this week, ni minggu keberapa? Ni minggu keberapa ni? Minggu keberapa? Minggu keempat. Macam mana setakat ni, minggu keempat ni? Rasa macam mana?
Boleh-boleh lah.
Boleh. ada tak siapa-siapa yang rasa burn out? Ada tak?
Ada sikit dekat bab 2.
Ada sikit lah dekat bab dia. Okay, faham. Okay, it's normal lah. Okay, for me it's normal to feel burn out ataupun ke semacam kenapa cepat sangat ataupun yelah macam banyak kerja, semua nak kena hantar semua, tapi it's okay. Macam sikit-sikit je macam dia sebab PDPR kan, PDPR dekat rumah, awak bukanlah sebagai seorang student, awak sebagai seorang anak, awak sebagai seorang kakak, awak sebagai seorang adik. So banyak role awak dalam rumah tu. Okay, sebagai seorang abang contohnya. So fahamlah kalau macam kalau contoh ada yang tak dapat join disebabkan internet ke ataupun tak dapat join sebab kongsi laptop ataupun kongsi phone dengan adik-beradik, faham je. Just boleh bagi tahu je saya. Okay, bagi tahu je apa kekangan yang menyebabkan awak tak boleh join. Okay. So saya harap SEM 2 nanti kita boleh berjumpa secara face to face. Okay, untuk SEM 2 nanti lagi senang. Alright, so these are the subtopic, the cell cycle, mitosis, meiosis. For this, alright, so we're going to discuss this subtopic, cell cycle, mitosis, meiosis for today, because for next week we're going to discuss genetic inheritance. Okay, genetic inheritance. So I'm going to cover everything in cell division, but only just surface. Okay, only C1, C2. Alright, so this is the cell cycle. Our objective for today is to show the stages of cell cycle. So I'm just going to show the stages and explain the stages in the cell cycle, which has interphase and mitotic phase. Okay, so for explain is C3, C4 punya level. So you're going to discuss it in your tutorial. Okay, so this is the cell cycle. This is the definition of the cell cycle, the complete sequence of events in the life of an individual diploid cell. Okay, complete sequence of events in the life of an individual diploid cell. So for example, we are diploid individual. Okay, we are diploid individual. Our chromosome number is 2n equals to, what is our chromosome number? Diploid chromosome number. For human, what is the diploid chromosome number for human?
46.
Anyone? 46. Thank you, Alia. Very good. Okay, it is 46. So we are individual diploid 46. So this is our cell cycle. So each cell in our body will undergo this cell cycle. And this cell cycle can be divided into two. We have interphase and mitotic phase, or we call that as M phase. Okay. If you refer to this cycle, you can see that the interphase is longest phase. Okay, the interphase is the longest phase. That's why when you draw the cell cycle, you must draw almost half of the circle. Okay, because this is the longest phase. Okay, this is when the cell will grow and prepare for cell division. Okay, it will grow in size. Okay, it will grow in size and prepare for cell division. It has three subphases. G1, we call that as gap 1. First gap phase, G1. 1 tu kena subscript tau. Dia tak boleh letak nombor 1 besar, subscript. Okay, as synthesis phase, and G2 is the second gap phase, or we call that as gap 2 phase. Okay, G1 as G2 phase. For the M phase or mitotic phase is the shortest part of the cycle. It includes mitosis and cytokinesis. The mitosis has 4 phases, which are prophase, metaphase, anaphase, and telophase. Pakmite. Okay, pakmite. Ingat pakmite. Okay, 4 phases. Prophase, metaphase, anaphase, telophase. Okay. Alamak, kena terang ke? Okay, saya terang sikit lah. Okay, saya terang yang highlight tu je. Okay, the one that I bold, I will discuss in lecture. Okay, G1 phase is this is when cell will grow in size. Okay, the cell increase in size, or we call that as cells growth. Okay, cytoplasmic volume will increase. Dia macam ni, daripada satu, sorry, daripada satu sel, dia nak hasilkan 2 sel. Okay, from one cell, it will produce 2 cells at the end. So in order to do that, it will need to grow in size. Okay, need to grow in size. Okay, cytoplasm dia akan banyak. Right, cytoplasm dia akan banyak. So when the cell is increased in size, the number of organelles will be also increase. Okay, the number of organelles will also increase, and it will build up energy storage. Okay, it will build up energy storage. That's why the one that we highlighted, the one that we bold in our in the lecture notes, is the important point. Okay, if the question asks you to state one of the events in the G1 phase, kalau awak tulis longest phase tak dapat markah. Okay, longest phase tak dapat markah. Kita bagi tahu yang penting dulu. Right, S phase, or we call it as synthesis phase. What we need to synthesize in this phase is the DNA. Okay, the DNA will replicate. This is when DNA replication occur. Okay, DNA replication occur. Okay, you will learn DNA replication in topic 6. Okay, you will learn in topic 6. And the importance in S phase, DNA is replicated to produce duplicated chromosome. Okay, to produce duplicated chromosome to ensure each daughter cell receive the same number and composition of chromosome as the parental cell. Supaya dia punya chromosome content tu samalah macam mak bapak dia. Okay, and then histone protein are synthesized so that the cells can make duplicate copies of each chromosome, and then to increase the amount of DNA. So these are the importance. Okay, we have 4 importance of DNA replication. Okay, to increase the amount of DNA. G2 phase. G2 phase, this is when energy store are increased. Masa G1 tadi, okay, G1, okay, G1. G1, this is when dia baru nak build up. Okay, dia baru nak build up energy storage. But at the end of G2, okay, at the end of G2, the energy storage is already increased. Okay, the energy store are increased. Okay. Then synthesis of microtubules, okay, for the development of spindle fiber. Microtubules is protein, okay, and centrosome and centriole will replicate. Okay, kalau tak ke dalam animal cell, okay, kita animal cell ada satu centriole saja. So bila dah dapat cell division, it will replicate and move to the opposite poles. Okay, just for that's not shown here. Just for not a fun fact, tapi ada just for your information, at this region, okay, at this region G1, at G1 phase, masa ni dia akan increase in size kan. Okay, the cell will grow in size. The cell will increase in size. Okay, if the cell is if the environment is not favor, okay, the cell will exit the cell cycle and it will enter G0. Okay, contohnya, okay, kalau dalam bahasa Melayunya saya cakap, dekat G1 ni, Kalau dia akan check tau, dia akan check environment ni okey ke tak. Maksudnya ada tak boleh tak dia proceed untuk masuk S phase? Boleh tak dia proceed masuk S phase? Dia akan check a few things. Contohnya cell increase in size kan? Cukup besar tak sel tu untuk masuk ke S phase? Contohlah, cukup tak protein, cukup tak makanan, cukup tak energi untuk masuk ke dalam S phase. Kalau tak cukup, bila dia tak cukup, dia akan exit the cell cycle. So when it exit the cell cycle, when it exit the cell cycle, it will enter G0. Okay, dekat sini dia tak akan proceed kepada S phase, dia akan keluar daripada cell cycle, it will enter G0. Kita panggil dia sebagai cell arrest, cell stop, cell arrest, dia tahan sekejap. Okay, cell arrest. Cell arrest, it can be hours, it can be days, or it can be years. Okay, it depends. Selagi dia tak cukup, dia tak akan proceed. Itu adalah salah satu cara untuk prevent cancer. Okay, untuk prevent cancer. Sebab cancer ni, cancer kita, siapa-siapa yang cancer lah, okay, you know cancer. Cancer is already common. Okay, cancer is already common genetic, okay, common genetic factor, common disease in our society. Okay, cancer tu disebabkan uncontrollable cell division. Okay, itulah. Alright, so that is banyak sangat cerita kan. Okay, that is interphase. Then kita pergi kepada mitotic phase. Okay, mitotic phase, we have mitosis. Okay, mitosis, it can be also called as karyokinesis, which refer to the division of nucleus. Okay, division of nucleus, which include prophase, metaphase, anaphase, and telophase. Okay, because this is mitosis, they will receive same number of chromosome. For example, if we same number of chromosome. So if we produce, okay, if our cell, our somatic cell is 2n equals to 46, okay, is our equal to n equals to 46, then we will produce two daughter cells which have same number of chromosome, also 46. Okay, cytokinesis refer to the division of cytoplasm. Cytokinesis, cytoplasm, okay, to form two cells, right? So kalau karyokinesis sampai sini je lah. Kalau tak ada cytokinesis, dia akan melekat macam ni. Okay, kalau tak ada cytokinesis, dia akan melekat macam ni. Kalau ada cytokinesis, which is the division of cytoplasm, then it will separate into two daughter cell. Okay. Alright. Okay. Human somatic cell contain 46 chromosome. Kita ada 46 chromosome. Kita lain kita human ataupun dengan kita dengan animal lain. Ayam mungkin ada 78. Okay, kucing mungkin ada 100 lain. Setiap individu, species or family of animalia, okay, dia punya chromosome content dia lain. Okay, unless that individual have mutation. If she or he has mutation, then the chromosome will be extra or less one, right? Every human reproductive cell or the gamete cell, sperm cell and egg cell contain 23 chromosome. So bila ada sperm 23, ovum ataupun egg cell pun 23. Okay, haploid. Two type of cell division. Occurs in somatic cell. Okay, mitosis occur in somatic cell. Semua sel dalam badan kita except for germ cell. Okay, it will produce two daughter cells. Okay, the number of chromosome in each daughter cell is equal to the number of chromosome of the parental cell. So, kalau parent dia 46, dia akan hasilkan dua daughter cell yang also 46. Okay, phomesis occurs in the gonad. Okay, saya tak suka guna gonad. Gonad ni salah Saya tak prefer guna gonad. Saya tak nak guna gonad. Germ. Okay, germ. As far as I'm concerned, the last time Last time we discussed among lecturers, we use germ cell, not gonad cell. Germ. Germ, G-E-R-M. G-E-R-M. I will confirm this later. Okay, I will confirm this with other lecturers later, and then I will update in our WhatsApp group. Okay, but I prefer to use germ. Okay, germ cell, germ, germ cell. Okay, it will for mitosis it will produce 4 daughter cell. Okay, the number of chromosome. Okay, the number of chromosome in each daughter cell half. 4 daughter cell setiapnya akan jadi half, iaitu haploid. Okay, kalau kita ada 46, start dengan 46, dia akan hasilkan 4 daughter cell yang haploid. So 23, 23, 23, 23. Okay, so mitosis. Describe the 4 stages of mitosis and the behavior of the chromosome for each stage. Describe briefly the cytokinesis process in animal and plant cell. Okay, saya kena explain benda dua ni dalam lecture. Okay, tak apa, saya akan explain, senang saja. 4 stages of mitosis and the behavior. Apa kromosom tengah buat dekat setiap stage. Describe briefly the cytokinesis process in animal and plant cell. Compare. Compare ni nanti awak discuss dalam tutorial saya tak pergi kepada C. Okay, mitosis. This is the definition. A division of somatic cell to produce 2 genetically identical daughter cell, with each cell having the same number of chromosome as the parental cell. Okay, the one that we bold in the lecture note is the keyword. Maksudnya awak tak boleh tinggal satu perkataan pun. Dia akan jadi salah. Okay. Walaupun awak dah ingat masa SPM ke apa, saya tak nak awak ingat benda tu dah. Ingat yang based on syllabus kita, which is this definition. Okay, produce 2 genetically identical daughter cell, which each cell having the same number of chromosome as the parental cell. So we have 4 stages. We have prophase, metaphase, anaphase, and telophase. This is prophase. Okay, prophase, metaphase, anaphase, and telophase. Okay, actually menarik lah kalau awak tengok bawah mikroskop. Okay, bawah mikroskop, kalau awak buat cell division, experiment 4, cell division you're going to see under microscope. Kalau awak buat cantik, awak buat pandai buat, memang nampak lawa macam ni. Okay, tapi itu untuk animal cell. Okay, prophase, what happened? What is the behavior of the chromosome in prophase? Okay, the spindle fiber form from centrosome. So this is spindle fiber, it will form from centrosome. Ester radiate. Ester tu macam spark. Ini adalah ester. Tu adalah ester. Ester tu macam spark tu. Itu adalah ester. Okay, centrosome move to the opposite poles, nuclear envelope and nucleolus disappear. Ini bukan lagi behavior. Behavior kromosom adalah point 1, 2, 3. Okay, dia tak cerita lagi pasal kromosom. Ini tak cerita. Yang 1, 2, 3 baru cerita pasal kromosom. So what happen is, during prophase, chromatin fibers will coil and condense. From this, it will become this. Coil and condense. Okay, it will be coil and condense. Chromosome shorten and thicken. Each chromosome consists of 2 sister chromatids joined at the centromere. So itu adalah itulah macam ni, macam butterfly tu kan. Okay, 2 sister chromatids joined at the centromere. Centromere tengah ni. Okay, that is prophase. Metaphase. Metaphase, this is when the chromosome or sister chromatids line up or arrange at the metaphase plate. Okay, the chromosomes or sister chromatids arrange or line up at the metaphase plate. Or you can call that as equator of the cell. Kalau yang tulis equator, you kena cakap equator of the cell. Tak boleh cakap equator. Equator tu boleh merujuk kepada garisan khatulistiwa bumi. Okay, sebab tu akan tulis equator of the cell. Garisan equator untuk sel. Alright, for anaphase, this is when the 2 sister chromatid separate and move towards opposite poles. So from here, this is sister chromatid, cantik, dia akan terpisah. Okay, terpisah. These two sister chromatids separate and move. Keyword here is separate and move. Dia separate dulu, terpisah dulu, baru dia bergerak. Move towards opposite poles with centromere move first. Each chromatid become an independent chromosome. Sekarang kita panggil dia sebagai chromosome. Okay, telophase. This is when the new nuclear envelope form. Okay, new nuclear tadi dah breakdown masa prophase. Okay, the new nuclear envelope form around each group. Nucleoli reform. Kita tak panggil form tapi kita panggil reform or reappear. Okay, nucleoli reform or reappear. The chromosome arise at the opposite poles. The chromosome will uncoil and lengthen. Okay, the chromosome will uncoil and lengthen. Okay, dia macam jadi bentuk berbenang balik lah. Okay, it will become less condensed, and return to chromatin form. Okay, prophase, metaphase, anaphase, telophase. Okay, cytokinesis in animal cells. So after telophase, it will proceed to cytokinesis. Okay, cytokinesis. Cytokinesis, we have cytokinesis are different. Okay, cytokinesis are different in animal cell and in plant cell. So in animal cell, the process, okay, it occur by process known as cleavage, in the appearance of cleavage furrow. Okay, process tu, dia akan form cleavage. Proses cytokinesis tu, kita nampak cleavage. Maksudnya kita nampak dia punya lekuk. Okay, so nanti dia akan form cleavage furrow. Okay, lekuk yang masuk ke dalam. What happen is the membrane is pulled inwards by the cytoskeleton. Cytoskeleton ni duduk dalam sel. So dia punya membrane cytoskeleton, dia akan tarik membrane. Bila dia tarik membrane, it will form a contractile ring. The contractile ring, which consists of actin and myosin. Okay, it will form on the cytoplasmic side of the cleavage furrow. Okay, and then the cleavage furrow will deepens. Okay, dia akan lekuk tu makin lama makin ke dalam. Bila makin ke dalam, it will reduce the diameter of the ring and it will pinch off. Okay, it will pinch off. The cells are separated completely and two daughter cells are formlah ni. Okay, dia formlah jadi macam tu. Okay, dapatlah dua daughter cell. So kalau cytokinesis in animal cell, cleavage furrow. Okay, cleavage furrow. In plant cell, formation of cell plate. Okay, formation of cell plate. Okay, ini adalah cell plate. Cell plate forming a new cell wall. Okay, formation of cell plate. So what happens is, the process begins, the vesicle derived from Golgi body. Okay, vesicle derived from Golgi body move along microtubules to the middle of the cell. Okay, so what happens is, Golgi body, salah satu function of Golgi body is to produce a vesicle. Okay, dalam vesicle tu adalah dia punya content. Okay, so what happens is, this vesicle, it will move along the microtubule. Okay, move along the microtubule to the middle of the cell. Okay, yang ni. Pergi ke tengah ni, carbohydrate filled vesicle. Okay, dia akan pergi kepada ni, tengah ni. Dalam vesicle tu, dia ada carbohydrate. Okay, dah ada carbohydrate. Carbohydrate yang untuk form cell plate. Okay, carbohydrate ni yang akan membentuk nanti cell plate. Okay, so what happen is, after the vesicle move along, dia macam ada satu track tau. Dia macam ada satu track. So dia ada satu track, lepas tu vesicle datang, dia follow je track tu. Okay, dia follow je track tu. The vesicle then, it will enlarge. Okay, dia follow, dia ikut je, the vesicle will enlarge and fuse to form a cell plate across the equatorial of the cell. Okay, and then cell plate will expand outward. Okay, lepas tu cell plate tu akan expand outwards and fuse with plasma membrane. And new cell wall form between the membrane and forming two cells. Lepas tu dapatlah dua sel. dapat sel wall yang baru, dia akan form 2 sel. Okay, itu adalah cytokinesis in plant cell. Okay, kalau animal cell cleavage furrow, kalau plant cell subplate. Alright. Okay, for compare you will discuss in your lecture. Okay, setakat ni okey ke? Apa okey kat tangan? Yang kat tangan kan? Alright, good, nice. Okay, bagus, thank you. Okay, Alhamdulillah. Okay, kalau kurang sikit faham, kalau kurang faham boleh tanya lecturer tutor awak. Okay, hebat-hebat lecturer tutor awak ni. Okay, kita pergi kepada seterusnya iaitu meiosis. Okay, meiosis. Okay, so I'm going to discuss this thing only. State the stages of meiosis 1, meiosis 2. Okay, objective define chromatid. Okay, defined chromatid, homologous chromosome, synapsis, bivalent, tetrad, chiasma, crossing over, and centromere. Term-term ni yang siapa yang selain yang SDS lah. Okay, yang baru, yang bukan SAS bukan SAS yang 3, saya tanya SDS. Term-term ni dah discuss ke masa SPM? SDS?
Dah, madam.
Dah ya. Oh, okey. Ada yang belajar bahasa Melayu ke? Ada yang belajar bahasa Melayu ke term-term ni?
Ada.
Ada. Okey, kalau kromatik panggil apa? Yang belajar bahasa Melayu ni.
Kromatik
Kromatik lah. Kalau homologous chromosome? Homologous chromosome?
Homolog.
Kromosom homolog. Synapsis? Thank you Zuhaili, thank you. Kromosom homolog. Synapsis? Kalau synapsis panggil apa? Sinapsis. Synapsis panggil apa? Kita brief dulu sebab ada kawan awak yang belajar bahasa Melayu. So saya nak translate dulu apa term dia. Kalau synapsis ada tahu tak? Kan synapsis juga. Kau awak tak tahu synapsis apa?
Cikgu saya panggil synapse je.
Synapse. Okey, boleh. Synapse itu dah jadi benda lain. Okey, tak apa. Okey, ada cikgu panggil synapse. Okey, tak apa. Kita ikut. Bivalent?
Bivalent juga. Semua je.
Bivalent. Okey, tetrad?
Tetrad juga.
Tetrad juga. Okey, chiasma.
Chiasma.
Okey, sama aja crossing over. Pindah silang.
Haa.
Ya.
Haa.
Oh, okey. Betul. Pindah silang. Centromere? Centroma. Okey. So itu adalah term-term bahasa Melayu. Kita akan guna bahasa Inggeris. So you need to define chromatid homologous chromosome. Thank you Izzati. Define chromatid homologous chromosome, synapsis, bivalent, tetrad, chiasma, crossing over, and centromere. State the stages of meiosis 1 and meiosis 2. Explain the behavior. Explain behavior ni awak akan discuss dalam lecture tutorial lah Okay. Meiosis, the definition is the division of germ cell. Ni saya cakap ni. Okay, the division of germ cell which produce 4 daughter genetically non-identical. Tak sama. Okay, anak dia tak sama muka. 4 daughter genetically non-identical. Dah lah tak sama. Lepas tu haploid daughter cell. 4 daughter genetically non-identical haploid daughter cell. Each having half the chromosome number of parental cell. So kalau parent 46, macam kita human parent 46, gamete akan jadi 23. So it occurs in germ cell. Okay, germ cell. Dia berlaku dalam germ cell that located in reproductive organs. Okay, all gonad. Sebab itulah kita tak guna gonad. Okay, macam tadi slide tadi kan, kita cakap occur in gonad. We don't use gonad because in gonad tu besar. Okay, gonad tu besar, dia punya tempat. Kat mana? Ada sel yang ada yang sel buat mitosis, ada yang sel buat meiosis. So dia jadi macam tak tepat. So better if the question asks you where does meiosis take place, in germ cell. Okay, so meiosis, it will produce gamete, which are sperms and ovum. Okay, sperms and ovum. Each daughter cell has half the chromosome number as parental cell. Okay, kalau parental cell diploid 2N, daughter cell akan jadi haploid. Contoh kita 46, daughter cell 23. Kan? Betul tak? Ke kena guna calculator? Betul tak?
Betul.
Okay, pandai. Thank you Izzati. Thank you Izzati, teman saya. Tak cakap seorang. Okay. Kalau ayam 38, dia punya parental cell, daughter cell akan dapat berapa? Ayam 38 parental cell, daughter cell haploid will be
19.
19. Very good. Thank you, Maizatul Ain. Okay. So meiosis, it will causes genetic recombination. Sebab itulah awak dengan adik-beradik awak unless twins. Ada twin tak dalam ni? Ada kembar tak dalam ni? Ada tak?
Ya.
Oh, siapa ni? Anas? Apa nama? Anwar. Oh ya? Anwar, Muhammad Anwar bin Nur Aziz. Kembar matrik juga ke?
Ya, KV, Banting.
Oh, Banting.
Perempuan.
KM Kolej Mara Banting?
Bukan college vocational. Dia perempuan.
Oh, perempuan. Kolej vocational. Oh, okay. Oh, lain lah. Dia lain lah. Okay. Meiosis causes genetic recombination. Sebab itulah awak dengan adik-beradik awak lain. Unless you are identical twin. Okay, you are identical twin. Macam siapa Siapa Tak boleh. Siapa twin dalam Malaysia ni? Kat tepi.
Anak Dr. Syed Muzaffar.
Tapi anak Dr. Syed Muzaffar. Anak Dr. Syed Muzaffar. Tapi bukan yang baru ni, bukan anak dia yang baby tu. Noah dengan Adam tu, kan anak dia lelaki dua tu. Okay, sama muka, almost sama lah muka dia kan. Anak Syed Muzaffar, betul anak astronaut. Okay, kembar. Okay, meiosis ni dia akan cause genetic recombination lah. Okay, dia tak sama. Okay, tak sama kita antara adik-beradik pun tak sama. So involve DNA it involve DNA replication during interphase 1 only in the parental cell. We have to, nanti kita cerita ataupun tutorial awak akan cerita kat mana dia ada genetic recombination ni, masa bila. We have two cycles of cell division, meiosis 1, meiosis 2. First meiotic division and second meiotic division. It occurs during meiosis occur during gametogenesis, which is the formation of sperms and eggs, in animal, and formation of spore in plant. Okay, dekat plant pun ada meiosis, formation of spore. Alright. Tak apa. Term. Okay, kita nak cerita pasal term. Terminologies in cell division. Okay, terminologies. Homologous chromosome. English, bahasa Melayu. Izzati apa? Apa tadi? Kromosom homolog Izzati?
Kromosom homolog juga.
Kromosom homolog juga. Okay, kromosom homolog. Okay, homologous chromosome. Homologous chromosome refer to chromosome that have same shape, size, and centromere position. Apa yang dia sama adalah same shape, size and centromere position. Satu daripada mak, satu daripada ayah. Contoh kita tengok yang ni. Okay, homolog. This is homologous chromosome. Human have 23 sets of homologous chromosome. Each set is made up of 2 homolog. So, yang ni lah, ini adalah homolog. Sama size dia. Shape, structure, centromere position dia pun sama. Maksudnya kalau dia duduk sama tinggi, diri sama tinggi, duduk sama rendah. Macam itulah. Dia punya partner lah. Homolog ni macam dia punya partner. Dia punya partner. Mesti, contoh kalau nak kahwin mesti sekufu. Contoh. Kahwin mesti sekufu. Macam ni lah. Okay, satu, dia kena sekufu, dia kena sama. Size, shape and centromere position. So kalau kita tengok ni, the 23 sets of homolog chromosome, they are the same. Okay, they are the same. Okay, and ni masa bila? Ini sebelum prophase. Sebelum prophase 1, dia memang duduk berterabur macam ni. Okay, dia tak jumpa lagi partner masing-masing. Okay, dia scattered. Once prophase 1 start, then they will pair up together. Okay, pasangan yang sama tinggi, sama rendah, sama shape, sama size, dia akan duduk sebelah-sebelah. Later kita tengok. Okay, chromatid. Chromatid is a thread-like strand formed from a chromosome. Okay, thread-like strand formed from a chromosome. This is chromatid. Each chromosome divides along its length into two sister chromatid. Each chromosome divide along its length into two sister chromatid. Yang ni adalah sister chromatid. Sama. Sister chromatid tu maksudnya dia share centromere yang sama. Okay, dia share centromere yang sama. Ya, ni adalah sister chromatid. Kalau saya tunjuk yang ni, contoh saya tunjuk yang ini dengan yang ini, itu adalah non-sister chromatid. Okay, dia tak sama. Okay, chromatid is thread-like strand from a chromosome. Each chromosome divide along its length into two sister chromatid, and the two chromatids are the exact replica of each other. Okay, dia macam twin. Sebab itu kita panggil sister. Okay, sister chromatids. Centromere. Okay, centromere. Centromere refer to the tak tunjuk. Okay, centromere refer to the ni center lah yang tengah-tengah ni. Okay, dia punya macam pusat lah, dia macam pusat. Okay, kat situ tengah-tengah. Center. Okay, the centromere is the constricted narrowest region joining the two sister chromatid. Yang ni lah, besar, kita tunjuk yang ni lah, centromere. That is centromere. Okay, it attached to spindle fiber during cell division. So it will attach to spindle fiber during cell division. Kalau tak attach, tak boleh lah. Dia tak akan jadi proses, maksudnya dia attach masa metaphase kan. Okay, masa metaphase tu dia dah attach. Kalau dia tak attach, dia tak boleh proceed kepada anaphase. Dia tak boleh proceed, dia akan floating around. Dia akan make sure the spindle fiber is fully attached to centromere, then it will proceed to the next phase. Okay. Okay. Synapsis. Synapsis refer to the pairing of homologous chromosome. Okay, pairing of homologous chromosome during prophase 1 and meiosis. Okay, so synapsis ni adalah proses yang di mana homolog kromosom akan duduk yang sekufu tadi. Okay, duduk dengan partner dia yang sama tinggi, sama same size, shape and centromere position. Synapsis is the process of the pairing of homologous chromosome during prophase 1. So before prophase 1, it will looks like this. So this is before prophase 1. Then when prophase 1 start, it will move towards the homolog chromosome. Okay, the homolog chromosome will pair with each other. Okay, dia akan duduk. Dia akan bentuk, at the end dia akan bentuk macam ni. Okay, yang tinggi duduk tinggi, yang rendah duduk dengan rendah. So that is pairing. Okay, pairing of homologous chromosome. So kalau kita tengok kat sini, ada homologous chromosome kan? One pair, ada satu. Okay, kalau kita tengok dalam kalau satu sel besar ni, kita ada dua pair of homologous chromosome. Dua pair. Pair pertama, pair kedua. Dua pair of homologous chromosome. Okay. Bivalent. Bivalent ni sama macam tetrad. Okay, dia adalah dia benda yang sama. Okay. Maksudnya dia refer kepada subjek yang sama. contoh kalau kita tengok bivalent, bi ni 2. Dia tunjuk kepada 2. So satu yang ni, ini adalah bivalent. 1, 2. 1, 2. So ada 1 bivalent. Okay, ada 1 bivalent. Okay, the structure formed when a pair of homologous chromosome is in synapsis state. So kalau kita tengok sini ada 1, 2, 1 bivalent. Kan kita tengok kepada tetrad. Tetrad ni tetrad ni adalah 4. Kan tetrad 4. So 4 ada 4 kaki. 1, 2, ada 4 kromatik. 1, 2, 3, 4 kromatik. 1 tetrad. Kita tak panggil 4 tetrad. Kalau 4 tetrad maksudnya ada lagi banyak lah. 1 merujuk kepada satu tetrad tu dah merujuk pada 4 kromatik. Sebab tu kita panggil 1 tetrad. A structure where a pair of homologous chromosome in a synapsis state that consists of 4 chromatids. Contohnya, kalau saya tunjuk kat sini gambar ni, this gambar, this diagram, one pair of homologous chromosome. Okay, kita tengok kat sini. Kat sini ada 2 pair. Sini ada 2 pair of homolog chromosome. Ada berapa bivalent? Ada berapa bivalent? 2 bivalent. Ya. 2 homologous chromosome, 2 bivalent, ada berapa tetrad?
2 juga.
2 juga. Okay, 2 tetrad. Okay, contohnya kalau kita tengok. Lah tak ada lah pula. Okay, contoh saya lukis. Contoh yang saya lukis sekejap. Okay, contoh saya lukis satu sel. Oh, faham je lah Saya tak bawa pen tertinggal kat rumah. Rushing, okay. Sekarang ada berapa homologous chromosome? Ada berapa pair homologous chromosome? Ada berapa pasangan homologous chromosome? Thank you. Ya, yeah, Izzati cakap berapa Izzati?
3.
Yes, Izzati cakap 3, Zuhaili cakap 3. 3 homologous chromosome. Berapa bivalent?
3.
3 bivalent. 1 bivalent pertama, bivalent kedua, bivalent ketiga. 3 bivalent. Berapa tetrad?
3.
Thank you Zuhaili, thank you Izzati. 3 juga tetrad. 3 tetrad. 1, 2, 3. 3 tetrad. Okay. 3 pair homologous chromosome, 3 bivalent, 3 tetrad. Okay. Tetrad tu dah menunjuk kepada 4. Tetra. Tetra tu menunjuk 4. So ada berapa? 1, 2, 3, 4. 1 tetrad. 1, 2, 3, 4. Tetrad yang kedua. 1, 2, 3, 4. Tetrad yang ketiga. Okay. Dia penting. Term ni awak perlu faham. Penting untuk awak faham. Sebab dia selalu, dia akan, macam-macam dia bentuk soalan dia boleh tanya. Dia boleh suruh awak label. Okay. Dia suruh awak boleh awak label. Dia suruh boleh awak lukis. Okay. So kena faham konsep tu dulu. Okay. Crossing over. Crossing over adalah proses. Okay, proses. Ini adalah salah satu proses lah. Selain synapsis tadi. Crossing over adalah proses of exchange of genetic material between non-sister chromatids during prophase 1. Pertukaran genetik daripada macam ini jadi macam ni. Okay, exchange of genetic material between non-sister chromatids. Okay, between non-sister chromatids. Okay. Between non-sister chromatids of homologous chromosome during prophase 1. So ini berlaku semasa prophase 1. Okay. And the site, tempat yang berlaku crossing over tu, kita panggil sebagai chiasma. Crossing over adalah proses. Region tempat yang berlaku crossing over tu, kita panggil sebagai chiasma. Chiasma singular, chiasmata plural. Chiasma singular, chiasmata plural. If we look at this diagram, okay, it is labeled as chiasmata. Okay, because it refers to two region of crossing over. So chiasmata. Okay, chiasmata. Chiasma singular. Okay, this is not included in your lecture note. Okay, this is not included in your lecture note, but just I'm going to explain. Okay, apa dia lah, sebab soalan ni pernah keluar. Soalan ni pernah keluar past year, tak silap saya. Okay, so ni kalau dia share same centromere, it is sister chromatids. Okay, if it is the same size, shape and centromere position, it is known as homologous chromosome. Okay, not same size, not same shape, not same centromere position, it is called as non-homologous chromosome. Not sharing the same centromere, it is called as non-sister chromatids. Okay, so make sure you know that, right? Term, okay, the term. Okay, for mysis 1, okay, for mysis 1 and mysis 2, okay, we have another 10 minutes. I'm not going to discuss in details. I'm going just to overview mysis 1 and mysis 2. Okay, mysis 1, we have interphase. Okay, first, of course, we have interphase. Then we have prophase 1. Prophase 1 is one of the important phase for mysis because most of the important events occur in prophase 1. Okay, in prophase 1, we have another 5 subphase. Okay, bawah prophase 1 ni ada lagi 5 subphase, iaitu lactotin, zygotin, pakitin, diplotin and dikinesis. Okay, lactotin, zygotin, pakitin, diplotin, dikinesis. Alright, and then we have metaphase 1, anaphase 1. Mysis 1 refer to separation of homologous chromosome. Okay, mysis 1 adalah kita nak separatekan homologous chromosome. Okay, yang pair tadi, yang couple tadi, kita nak separatekan couple tu. Okay, macam SEM 1. SEM 1 lah, mysis 1 ni macam SEM 1. Ada couple. At the end of SEM 1, kita nak separatekan couple tu. Nak separatekan homologous chromosome. Okay, mysis 1. Mysis 2 is the separation of sister chromatids. Okay, mysis 2 is the separation of sister chromatid. If you look at metaphase 2, we will separate, okay, metaphase 2, anaphase 2, we will separate the sister chromatids. Okay, and at the end, at telophase 2, we have 4 daughter cells. Okay, 4 daughter cells. Each of the daughter cells has haploid chromosome number. Okay, so this is example of mysis that takes place in the germ cell. Okay, mysis that takes place in the germ cell. Right? We're going to learn this in topic 9. Okay, topic 9, reproduction. Right. All right. I think that's all for lecture today. Do you have any questions? Do you have any questions? Do you have any questions? Zohaili got no question. Do you have any questions, Elaine? All right. So we will end our lecture now. Okay, I'm going to share recording later. And okay, right now we have 81. Okay, 81 out of 83. 81 saya masukkan 2, so 79 dah lah daripada 83. Okay, alright, thank you class. If you don't have any question, I will end our class now. I will stop our recording. And then untuk lagi berapa minit ni? Okay, lagi 7 minit macam tu bolehlah pergi minum kejap ke, pergi makan kejap ke. Okay, so untuk next class. Alright, thank you class. See you next week. Assalamualaikum. Bye-bye.
Waalaikumsalam.
Salam.
Thank you, Madam.
Thank you,
Thank you,
Madam.
Thank you.
Thank you, Madam.
Madam.
Sini. Thank you. Thank you, Zoili. Thank you. You have a nice day. Have a nice day, guys.